Hello, everyone. My name is Aruna, and I'm currently a fourth-year undergraduate at UC Davis. And today I'm presenting with Sid, who's a second-year PhD student, and we both work at the Quan Lab. And we're really grateful to be here today and be able to present our research with all of you. And today we're talking about predicting neurologic disorder status with brain somatic mutations. Neurologic disorders affect approximately 1 in 3 people globally. And one of the hardest things to understand about them is understanding the genetic factors that cause these disorders to happen. That's because most of these disorders are polygenic, meaning that there's more than one mutation that's causing them to happen. Past work in this field has looked at mostly inherited mutations. An example would be GWAS that many people presented about today, genome-wide association studies, mainly trying to understand inherited mutations that cause these disorders to happen. But our work is different because we're focusing on somatic mutations, and somatic mutations occur post-zygotically, and they're not inherited or passed down. And so mutation that occurs earlier on in development will be more prevalent in the person throughout their life. So in cancer genomics, there's this framework called clonal trees, where they're able to predict the frequency, or they're able to predict when a mutation occurs depending on its frequency relative to a tumor. And our goal, similarly, is to understand when a somatic mutation is occurring, but not in reference to a tumor, but in reference to the development of the individual themselves. And specifically, we want to understand the difference between how these trees would look like between cases and controls. But one drawback that we have is that we're not working with as many mutations as many of the cancer genomic studies are working with. They have around thousands of mutations, whereas our work is near the hundreds. And so we're trying to develop a framework that can understand the difference between what these trees would look like if we were able to rebuild them properly, but without actually having to rebuild the trees. And so we're testing out 2 separate hypotheses. The first one is that there is a systematic difference between the structures of trees inferred by somatic mutation frequencies for cases and controls. And the second hypothesis is that there's a systematic difference in regulatory functional impacts of somatic mutations between cases and controls. And to test these hypotheses, we are building a transformer-based classifier architecture. So this is the input into our model. We're aligning the mutation frequencies in decreasing order, and this was to represent how they could have been inherited by the person, higher frequency meaning inherited earlier on in development, lower frequency meaning inherited later on in development. And then they are projected to 10 dimensions. This goes into the transformer encoder, and the output is a classification of a case control status. If you notice, we are not using a positional encoding currently, but initially to test our hypothesis, we did incorporate the positional encoding, but it didn't work the way that we wanted to. And this is our input into, or these are all the studies that we're using. We have 3 different disorders that we're looking at: autism spectrum disorder, Tourette syndrome, as well as schizophrenia. And when training the model, we're training them, all 3 of them separately. And 98% of the mutations that we're working with are from non-coding regions. And so one of the drawbacks, as I mentioned earlier with our data, is having a small number of mutations for each of the samples. And we also have a small number of samples in terms of cases and controls as well. That's because many of these, identifying somatic mutations requires having deep sequencing of postmortem brains, which is difficult to acquire. And so now we'll be talking about how we overcame these problems as well as talking about our results.
So to ensure robustness of our modeling, with the small data set size and the small number of mutations per sample, we ensured that we wanted to run this data over multiple random splits of 10-fold stratified cross-validation. And we wanted to measure the average validation AUROC across folds per split. And the second thing we wanted to do was we keep the input the same and train another model by shuffling the output labels. And so if the model does better on the non-shuffle data, that's the real data, versus shuffle data, that means that what we're learning is robust signal. And so we can see the results here. On the y-axis, we have the average validation AUROC for the not shuffle data. And on the x-axis, we have the validation AUROC for the shuffle data. So each data point has the y value from the model trained without shuffling and the x value for the validation AUROC from the model with shuffling. And we would hope that all our data points fall above the a diagonal dashed line, which is the y equals x line. And even more so, validation AURC expected result is zero point 0.5, which is by chance. So we would hope that our validation for our real data is above zero point 0.5 and probably lower than zero point 0.5 for our shuffle data. So we would hope that all our points are in the top right quadrant here above the dashed line. And first we see for our first hypothesis a negative result, but mutation frequency is not predictive of disorder status. So the points here, as we can see for 10 different splits, do not lie all above the y equals x line and not in the upper left quadrant. And so we don't have enough information to reject the null hypothesis that there is no structural difference between case control trees inferred by only mutational frequency. So then we wanted to look at our second hypothesis, which was assessing the functional regulatory impact of these somatic mutations. And as mentioned before, about 97 to 98% of our mutations were non-coding. And so we wanted to look at the non-coding functional regulatory landscape in two independent ways. Number one, to ATAC-seq for the chromatin landscape, and number two, histone modification chip-seq for the epigenomic landscape. And so we have our chromatin accessibility ATAC-seq data from the catalyst or the chromatin accessibility atlas on a single cell level, and we have our histone modification data from the roadmap epigenome project. And so we have different cell types for the signals that we have for these different modalities. And we have variants that sit on, as we can see here, at different positions with respect to these signals. And so the way that we wanted to encode them is whether or not a mutation intersects a signal peak. So whether it does, then there's a 1 or a 0 if it doesn't for each of these cell types. That's how we input that into the same transformer architecture that we were talking about. And we did training again to predict case control status. And now we see here some positive results, which is that ATAC-seq data across these 143 cell types that we looked at is predictive of disorder status across our ASD cohort, our TS cohort, and our schizophrenia cohort. And we see that the 5 splits that we used here for the same kind of plots I explained about before, we see that the dots are above the y equals x line and towards the top left quadrant of the graph, as we would hope. And then we look at orthogonally, after testing a bunch of histone modifications, we looked at H3K4ME1, which is known to be associated with active cis-regulatory elements, promoters and enhancers. And so we did the same kind of embedding, whether the mutations intersect the peaks of these signals or not. And we got results that we see that, that there's robust signal indicating that we can predict case control status from these inputs. And so we wanted to see why the model does so well, like why we're able to get such distinctions between case and control status from these inputs. And so we looked at feature attribution, which is going backward from the output to the input, to see which features in the input kind of drove output prediction or classification. And so we have these feature attribution heat maps, which is like this one is for just one sample. And on this axis, the x-axis, we have like the individual variant. And I know it's illegible, but bear with me. There's the somatic mutation is here. And then on the y-axis is the different cell types for the data. And so we wanted to try to visualize all of the samples across the studies and see if there's differences in cases and controls based on the attributions that would tell us that the model is learning something based on these cell types for cases versus controls. And so we decided to cluster the cell types based on hierarchical clustering of the PCAs of the input. embedding, and then we chose five. We chose to cut the tree such that we had five clusters, and they correspond to mainly neuronal, mesenchymal, epithelial, mixed, and hematopoietic cell types. And then we wanted to collapse further across all variants in an individual that have non-zero attribution scores to compare if there was some kind of cell cluster specific impact of these mutations, different between cases and controls. And so then we wanted to put them all together to compare all the samples as single columns now in this heat map. And that's what we have here. So we see that these average attribution scores show case control differences. So we have on the top here, the ASD cohort with 73 individuals. So each column is an individual sample, and each of the rows corresponds to like the cell cluster that we were talking about. And so on first glance, we can see that there is a difference in the pattern between cases and controls for this heat map. What is not yet so apparent is like there's cell cluster specific differences. That would be like each row in the heat map here would be like different between cases and controls. We can see like an overall larger score or like a red pattern in cases versus controls, at least for the open chromatin ATAC-seq data. And to a lesser extent, we can still see some differences between cases and controls for the H3K4ME1 data. And it's interesting then, we look at the schizophrenia cohort with only 24 individuals and samples, and then we can still see case and control differences, but it seems that for the ATAC-seq data with open chromatin information versus the H3K4ME1 data, we get like opposing results because we can see that there's like the cases are kind of like less red or there's less attribution here than controls, but it's swapped kind of in the H3K4ME1 data. So we need to look more closely at possibly like the different sets of mutations or variants that the model is looking at for each of these data sets. But our conclusion is that we still see that the attributions show learned differences between cases and controls from a model. And that leads me to our discussion. So through our transformer classifier model, despite the small data set size, which is currently the largest data set we could get for somatic mutations, we see one, no signal to indicate systematic differences in tree structures between cases and controls. That's failed hypothesis one. But two, strong signal from independent ATAC-seq and H3K4ME1 data to indicate systematic differences in non-coding regulatory functional impact between cases and controls. And further, even though hypothesis one failed, it still provides information that our results from hypothesis two are not confounded by differences in just mutational frequencies between cases and controls. And with that, I would like to acknowledge our principal investigator, Dr. Gerald Kwan, and the Kwan Lab members for supporting us in this project, and the NIH for funding our research. Thank you.
I think it's very hot, and I'm curious about the somatic lines of mutation going up in the floating regions, why is it going to gene regions or floating regions? And then what's what is conditions?
Okay, so the data set that we're working with specifically had non-coding regions. So it's not that it's not occurring in coding regions as well, but we were specifically wanting to look at non-coding somatic variants.
Yeah, and in terms of like the origin of these somatic variants, it's still unclear as to what causes them. There could be errors in DNA replication and other things. And so the data that we had is, again, very little, as we said, but in that most of our variants are in the non-coding space.